How are vaccines made? Vaccines have long been hailed as one of our greatest public health achievements. They can be made to protect us from infections with either viral or bacterial microbes. Measles and smallpox, for example, are viruses. Streptococcus pneumonia is a bacterium that causes a range of diseases, including pneumonia, ear and sinus infections, and meningitis. Hundreds of millions of illnesses and deaths have been prevented due to vaccines that eradicated smallpox and significantly reduced polio and measles infections. However, some misunderstanding remains regarding how vaccines are made and why some scary-sounding ingredients are included in the manufacturing process. The production of our vaccines has evolved a lot since the early days, when vaccination was potentially dangerous. Inoculating an individual with ground-up smallpox scabs usually led to a mild infection, called variolation, and protected them from acquiring the disease the regular way, via the air. But there was always a chance the infection could still be severe. When Edward Jenner introduced the first true vaccination with cowpox, protection from smallpox became safer, but there were still issues. The cowpox material could be contaminated with other germs, and sometimes was transmitted from one vaccinated person to another, leading to the inadvertent spread of blood-borne pathogens. We've come far in the last 200 years. There are different kinds of vaccines, and each requires different processes to move from the laboratory to your physician's office. The key to all of them is production of one or more antigens, the portion of the microbe that triggers a host immune response. There are several methods to produce antigens. One common technique is to grow a virus in a cell culture, typically grown in large vats called bioreactors. Living cells are inoculated with a virus and placed in a liquid growth medium that contains nutrients, proteins, amino acids, carbohydrates, essential minerals, that help the virus grow in the cells, producing thousands of copies of itself in each infected cell. At this stage, the virus is also getting its own dose of protective medicine, antibiotics like neomycin or polymyxin B, which prevent bacterial and fungal contamination that could kill the cells serving as hosts for the virus. Once a virus completes its life cycle in the host cell, the viruses are purified by separating them from the host cells and growth media, which are discarded. This is often done using several types of filters. The viruses are small and can pass through holes in the filter that trap larger host cells and cell debris. This is how live attenuated vaccines are created. These vaccines contain viruses that have been modified so that they are no longer harmful to humans. Some of them are grown for many generations in cells that aren't human, such as chicken cells, so that they have mutated to no longer cause harm to humans. Others, like the influenza nasal mist, were grown at low temperatures until they lost the ability to replicate in the warmer temperatures of the lungs. Many of these vaccines you were probably given as a child: measles, mumps, rubella, and chickenpox. Live attenuated vaccines replicate briefly in the body, triggering a strong and long-lasting response from your immune system. Because your immune system kicks into high gear at what it perceives to be a major threat, you need fewer doses of the vaccine for protection against these diseases. And unlike the harmful form of the virus, it is extremely unlikely, because they only replicate at low levels, that these vaccines will cause the host to develop the actual disease, or to spread it to other contacts. One exception is the live polio vaccine, which could spread to others, and, extremely rarely, caused polio disease. Approximately 1 case of polio from 3 million doses of the virus. For this reason, the live polio virus was discontinued in the United States in 2000. Scientists use the same growth technique for killed or inactivated vaccines, but they add an extra step: viral death. Inactivated viruses are killed, typically via heat treatment or use of a chemical such as formaldehyde, which modifies the virus's proteins and nucleic acids, and renders the virus unable to replicate. Inactivated vaccines include hepatitis A, the injected polio virus, and the flu shot. A dead virus can't replicate in your body, obviously. This means that the immune response to inactivated vaccines isn't as robust as it is with live attenuated vaccines. Replication by the live viruses alerts many types of your immune cells of a potential invader, while killed vaccines primarily alert only one part of your immune system, your B cells, which produce antibodies. That's why you need more doses to achieve and maintain immunity. While live attenuated vaccines were the primary way to make vaccines until the 1960s, concerns about potential safety issues and the difficulty of making them mean that few are attempting to develop new live attenuated vaccines today. Other vaccines aren't made of whole organisms at all, but rather bits and pieces of a microbe. The combination vaccine that protects against diphtheria, pertussis, and tetanus all at once is one example. This vaccine is called the DTaP for children and TDAP for adults. It contains toxins, the proteins that cause disease, from diphtheria, pertussis, and tetanus bacteria that have been inactivated by chemicals. The toxins are called toxoids once inactivated. This protects the host, aka you, potentially, from developing clinical diphtheria and tetanus disease, even if you are exposed to the microorganisms. Some viruses have toxins, Ebola appears to, for example, but they're not the key antigens, so they're not used for our current vaccines. As they do when developing live attenuated or inactivated vaccines, scientists who create these bacterial vaccines need some target bacteria to culture, but because the bacteria don't need a host cell to grow, they can be produced in simple nutrient broths by vaccine manufacturers. The toxins are then separated from the rest of the bacteria and growth media and inactivated for use as vaccines. Similarly, some vaccines contain just a few antigens from a bacterial species. Vaccines for Streptococcus pneumonia, Haemophilus influenzae type B, and Neisseria meningitis all use sugars that are found on the outer part of the bacteria as antigens. These sugars are purified from the bacteria and then bound to another protein to enhance the immune response. The protein helps to recruit T cells in addition to B cells and create a more robust reaction. Finally, we can also use genetic engineering to produce vaccines. We do this for hepatitis B, a virus that can cause severe liver disease and liver cancer. The vaccine for it consists of a single antigen, the hepatitis B surface antigen, which is a protein on the outside of the virus. The gene that makes this antigen is inserted into yeast cells. These cells can then be grown in a medium similar to bacteria and without the need for cell culture. The hepatitis B surface antigen is then separated from the yeast and serves as the primary vaccine component. Once you have the live or killed viruses, or purified antigens, sometimes chemicals need to be added to protect the vaccine or to make it work better. Adjuvants, such as aluminum salts, are a common additive. They help enhance the immune response to some antigens by keeping the antigen in contact with the cells of the immune system for a longer period of time. Vaccines for DTaP, Tdap, meningitis, pneumococcus and hepatitis B all use aluminum salts as an adjuvant. Other chemicals may be added as stabilizers to help keep the vaccine working effectively even in extreme conditions, like hot temperatures. Stabilizers can include sugars or monosodium glutamate, also known as MSG. Preservatives can be added to prevent microbial growth in the finished product. For many years, the most common preservative was a compound called thimerosal, which is 50% ethyl mercury by weight. Ethyl mercury doesn't stick around. Your body quickly eliminates it via the gut and feces. This is different from methylmercury, which accumulates in fish and can, at high doses, cause long-lasting damage in humans. In 2001, thimerosal was removed from the vaccines given in childhood due to consumer concerns, but many studies have demonstrated its safety. Finally, the vaccine is divided into vials for shipping to physicians, hospitals, public health departments, and some pharmacies. These can be single-dose or multi-dose vials, which can be used for multiple patients as long as they're prepared and stored away from patient treatment areas. Preservatives are important for multi-dose vials. Bacteria and fungi are very opportunistic, and multiple uses increase the potential for contamination of the vaccine. This is why thimerosal is still used in some multi-dose influenza vaccines. Though some of the vaccine ingredients sound worrisome, most of these chemicals are removed during multiple purification steps, and those that remain, like adjuvants, are necessary for the vaccine's effectiveness, are present in very low levels and have an excellent track record of safety.